Hello friends, today we are going to discuss about one of the important topic about urea cycle. Urea cycle, otherwise called Krebs Henseleit cycle. It is the first metabolic cycle to be discovered just 5 years before the that means TCA or Krebs cycle. Krebs Henseleit means it is attributed by the scientists who elucidated the cycle. In previous classes, we have discussed about transamination and deamination reactions, which are the common amino acid metabolic reactions. Transamination, the transfer of the amino group from amino acid to keto acid, which involves one pair of amino and keto acids mediated by pyridoxal phosphate, what we call PLP, an important coenzyme derived from vitamin B6. Then deamination, the liberation of the amino group from the amino acids in the form of ammonia is deamination. That means ultimately transamination and deamination leads to the ammonia, that means in the form of ammonium NH4 plus, because ammonia at optimal physiological conditions exists in NH4 plus. And where the fate of the ammonium leads to urea. Why? Because ammonia is the most toxic substances and that leads that have huge bearing on the body's physiological machinery. So it has to get detoxified. For that, urea cycle is an option. We are all ureotelic animals. Say mammals in general, human beings in specific are ureotelic animals. That means the chief or the major nitrogenous metabolic waste is urea. Such animals are called ureotelic. That means ammonia will get detoxified in the form of urea, and this urea through urine it will be excreted outside. In other organisms like birds, it is uric acid, so that is called uricotelics. Say in fishes, it is like ammonotelic. Here, ureotelic is an adaptation to terrestrial adaptations. If the water is too much low, then it will be uricotelic. If the water is too much high, then it will be ammonotelic, because to excrete ammonia, plenty of water is required. But ureotelic is not like that. So we are being terrestrial animals, we have to conserve the water and have to remove the chief nitrogenous metabolic waste in the form of urea. This is about the basic issues related to urea cycle. That means otherwise called Krebs Henseleit cycle, the discoverers who elucidated the urea cycle. Okay. Regarding other issues, urea, CO and H2 twice. This is a basic chemical structure of urea. We will discuss about, then before going to the cycle, we will discuss about some basic issues. Here, amino group, there are two amino groups. One amino group is coming from ammonia, other from aspartate, and the carbon atom is coming from carbon dioxide. This is very important. We have discussed the nitrogen content of the amino acids will be released in the form of ammonia. Right? We have discussed during our in previous classes that glutamate is a collecting source of N content, and this glutamate will subject to oxidative deamination, liberation of ammonia, then urea, then to urine. Okay, and this ammonia is the source of this one amino group, whereas another amino groups come from aspartate. I will tell you how this aspartate contributes in urea cycle. Okay, then carbon atoms come from carbon dioxide. From where? Actually there is an intricate relationship, a bicycle integrating relationship between urea cycle and the Krebs cycle. For instance, the carbon dioxide that releases in the Krebs cycle will be utilized in the urea cycle. At the same time, the ATP generated, say 12 ATPs in Krebs cycle, wherein 4 ATPs will be used in urea cycle. So there is and also the intermediate compound like fumarate released in urea cycle will link to the Krebs cycle, and also gluconeogenesis as well. So urea cycle and Krebs cycle are integrated. There is a intrinsic relationship, that what we call integrated pathways at different ways, like fumarate, carbon dioxide, and ATP. There are three links that connects the Krebs cycle and the urea cycle. regarding its basic issue. And most important thing is urea cycle actually is a 5-step cyclical reaction, cyclical metabolic cycle, wherein two enzymes belong to mitochondria, whereas three enzymes belong to cytosol. That means mitochondria and cytosol. Where it takes place is very important. We have already discussed. Urea is a source where our nitrogen metabolic waste will be excreted, and it takes place in liver. That means urea cycle takes place in liver. I will tell you why. Because most of the enzymes connected to urea cycle are present in liver. Though there are other tissues also where the enzymes relevant to urea cycle also present, but most important enzymes are present in liver. That is why it takes place in liver. Okay. Then it will reach to kidneys. Then through urine it will be excreted out. This is the pathway. The liver, the kidney and the urine. Out of which in liver, we have different cells. Two enzymes, that means it's a 5-step cyclical metabolic reaction. That means metabolic cycle wherein that means 5 enzymes. Out of which 2 enzymes will be present in mitochondria and 3 enzymes will be cytosol. So cytosol and mitochondria are the locations where the urea cycle could takes place. And there is, it's like regeneration. Just like in Krebs cycle we have oxaloacetate. It has to continuously get regenerated because oxaloacetate has to combine acetyl coenzyme A that forms citric acid, like that. Same mechanism here. Here ornithine occupies the role of oxaloacetate. Ornithine has to continuously regenerate it. Ornithine. So that the 5-step metabolic reaction will get will continuously synthesize urea that will be excreted outside, because ammonia is very toxic, so it has to get detoxified. So it will take place in here. So it's about the basic issues. Let us come to the cycle. Urea cycle. As we discussed, it's a 5-step metabolic reaction. Metabolic cycle, sorry. 5-step metabolic cycle. The first step include NH4 plus CO2 leads to carbamoyl phosphate catalyzed by the enzyme what we call CPS1, means carbamoyl phosphate synthase 1. Here 2 ATPs will be utilized. 2 ATPs. It's as usual reaction 2 ATP plus PA. N-acetyl glutamate. N-acetyl for activation, allosteric activation, N-acetyl glutamate. The thing is, CPS1, what we call carbamoyl phosphate synthase 1, will require NAG, what we call N-acetyl glutamate. That means for what? Allosteric activation is done by, that means allosteric activation of CPS1 is done by NAG. CPS2, another enzyme, that means carbamoyl phosphate synthase, but is present in cytosol. CPS1 is present in mitochondria. CPS2 present in cytosol. It is involved in the synthesis of pyrimidines, but here it involves in the synthesis of carbamoyl phosphate. This is very important. Carbamoyl phosphate. This is very important. Just imagine, for convenience for understanding, just like acetyl coenzyme in Krebs cycle, you can correlate for understanding. And this carbamoyl phosphate is the first reaction. This is the first reaction. It combines with ornithine. catalyzed by the enzyme ornithine transcarbamylase that leads to citrulline. That means this is the second reaction. I have already discussed. 2 reactions will be mitochondria, 3 reactions in cytosol. Up to this entire step will take place in mitochondria. Then to transporters, transporters. Transporter, the word impact. Just like we were discussing in previous class, membrane transistor now. The citrulline will come outside. That means out of the mitochondria into cytosol. It condenses with aspartate to form arginosuccinate. Catalyzed by the enzyme arginosuccinate synthase. Here ATP will be utilized in the way of, that means ultimately AMP plus PPI, and this PPI will later leads to inorganic phosphate. Okay, this is third reaction in cytosol. Okay, then after this arginosuccinate will again get into catalyzed by the enzyme arginosuccinase to split. That means actually arginine and fumarate, and this arginine catalyzed. This is the fourth reaction. Catalyzed by the enzyme arginase to split into urea and ornithine, and this all enter into mitochondria later. This is the final step. This is very important, and this fumarate will get into the Krebs cycle, for instance. So these are the 5-step reaction. That means 5-step metabolic cycle. I have already discussed. Urea is a 5-step metabolic cycle. In the first mitochondria, ammonium combines with carbon dioxide. That means it condenses to form carbamyl phosphate. Catalyzed by the enzyme CPS1, carbamyl phosphate synthase 1, whereas CPS2 present in cytosol for pyrimidine. But this CPS1 are actually rate dependent enzyme and depend on NAG. That means N-acetylglutamate for allosteric activation. This is very important. And this is irreversible. Of course, urea cycle is an irreversible cycle. Okay. And after the carbamyl phosphate, the ornithyl will combine with carbamyl phosphate to synthesize citrulline, catalyzed by the enzyme ornithyl transcarbamylase. It's the second mitochondrial reaction. That second step, what we call it. Up to this, it will take place in mitochondria. Then the citrulline to transporters will come out of the mitochondria into cytosol, and this citrulline combines with aspartate. We have discussed the second amino group coming from aspartate. This is the second source of amino group, because CO and H2 twice. CO and H2 twice. One amino group is coming from aspartate, one from ammonia. Okay, and this arginosuccinate. That means here one ATP will be utilized, and it tells us the AMP plus PPA. Here ATP to AMP means 2 actual ATPs. So 2 ATPs for synthesis of carbamyl phosphate and 2 ATPs here for synthesis of arginosuccinate. And here one amino group will be added. Arginosuccinate synthase. This arginosuccinate then catalyzed by the enzyme arginosuccinase splits into fumarate and arginine. This is the fourth metabolic reaction. This is third, and this is the fourth metabolic reaction. And this fumarate, I have discussed that there is a link between Krebs cycle and the Urea cycle. Through fumarate channel, through carbon dioxide channel, and the ATP. Carbon dioxide released in the Krebs cycle will be utilized here. And ATP, let's say 12 ATPs will be synthesized. The Krebs cycle will be utilized here, out of which 4 ATPs, Then fumarate. The fumarate will become malate, and malate will become acetoacetate. So Krebs cycle now. So it links Krebs and Urea cycle. Then the arginine, catalyzed by the enzyme arginase, again splits. That means arginase splits arginine into urea and the ornithine. We have discussed ornithine can be corroborated with oxaloacetate. It has to get regenerated. Just like ornithine also, it has to get regenerated for continuous, synthesis or generation of urea cycle. And this ornithine from cytosol will reach the mitochondria. Then again it will repeat. 5-step metabolic cycle. And this urea in the 5th step, look at this enzyme. Arginase is mostly present in liver. We have discussed. Urea cycle mostly takes place in liver. Why? Because most of the enzymes related to urea cycle will take place in liver. Say arginase mostly present in liver. Though there is a gradation of urea cycle takes place in other tissues, but liver is a hub. Okay, and after this, after we get the urea at the 5th step, utilized by the enzyme arginase. This is very important. They may ask you which enzyme is responsible for the release of urea. Arginase. So here arginine is the immediate precursor for ornithine. That means immediate precursor for urea actually. That means then of course it's split into ornithine as well. So ultimately urea is released out. So it is the immediate, that means arginine is immediate precursor for urea. And again the ornithine will be regenerated. This will be continuously, synthesized and repeated. And this urea will diffuse out of the liver through blood circulation, reach the kidneys, and through urine it will be excreted out. This is about the urea cycle. That means 5-step metabolic action, 2 in mitochondria and 3 in cytosol. And regarding the energetics, just I am writing here. How many? That means just as for understanding, we all discussed that NH4 plus, what's called energetics, plus CO2 plus aspartate plus 3 CO plus 3 ATPs, that leads to urea plus fumarate plus 2 ADP plus PI plus AMP plus PP, pyrophosphate. So totally 4 ATPs will be utilized in the urea cycle. They might ask you how many ATPs will be utilized in urea cycle. 4 ATPs. This is very important. And first important enzyme CPS1 is present in mitochondria. So it's about the energetics of urea cycle. Then I told you there is intricate relationship with Krebs cycle at different point, like fumarate point, carbon dioxide point, and ATP point. So and this through urea, that means ultimately the urine it will be released out, and it is the most common reactions in ureotelic animals because it is the chief nitrogen metabolic source. Okay, thanks for watching.